Are we still having these or no? Or are you gonna put it on?
I will
Gonna
put
put
it
it
on.
on.
If,
Okay.
if this is correct, I will put it like this.
Okay, yeah, that's correct. Sorry, I'm confused. Okay. So,
Okay,
the
you're still in the
concept,
shot.
I am?
You were a second ago.
Oh,
You're-
sorry.
you're good
Okay,
now.
okay, okay. So the concept we're covering today is DNA replication and how DNA fixes its errors during replication. In molecular biology, DNA replication is a biological process of producing one identical replica of DNA from one original DNA molecule. This process occurs in all living organisms and is the basis for biological inheritance. DNA is made up of a double helix of two complementary strands. So the process of DNA replication begins as such. An enzyme called helicase comes in. Here's helicase coming in, and it unwinds the two strands of the DNA, forming the replication forks. Following the rule of base pairing, two complementary strands are created. The DNA polymerase is able to replicate the leading strand in the 5' to 3' direction, as well as the lagging strand, but in a more discontinuous fashion. So both the lagging and the leading strands provide a template that attracts and binds complementary bases. Adenine with thymine, and guanine with cytosine. So as a result, a new DNA is created with one new daughter strand and one old strand. And that's why the process of DNA replication is called a semi-conservative replication.
Yay!
There are a few ways that a DNA can be damaged. It can be done either internally through some sort of a cell mutation. This is where a base pairing of the DNA sequence is off. It could be as little as just one letter off of the entire sequence, and that will create a mutation, or it could be done externally through any number of environmental factors. So how is the damage detected in DNA in the first place? So here we see a double-stranded, double helix DNA. The damaged DNA actually alters the spatial configuration of the DNA. So the DNA shape physically changes. So the one above may look something like this. Oops. So it does change the shape, and that's kind of how the change is depicted physically. And so what happens once the damage does occur, it recalls specific DNA molecules that bind at or near the site of damage. It could be anywhere really in this zone where the shape was changed physically. There are two mechanisms that are repair mechanisms for the DNA strand. So number one is single strand repair. And it is, as the name implies, it's fixing just one of the strands, which in this picture you can see that it's this strand that needs to be fixed here. And of course the second is, as you may be able to guess, the double strand repair mechanism. And as the name implies, again, it's really fixing both of the strands here, this one and this one. So there are three, within the single strand repair, there are three main types of repairs, and I will write them here. Number one is nucleotide excision repair. So I'll just put nucleotide. right here. So the nucleotide excision repair, and one of the best examples of that type of repair is a UV light damage. And so what happens is the pyrimidine dimers are recognized by the enzymes called endonucleases. And what they do, these enzymes, they go into the strand, into the cell, and they cut out the damaged nucleotides, which is how this name is implied here, nucleotide. Endonucleases, they cut out the damaged nucleotides. Then the DNA polymerase replaces the base, and the DNA ligase reseals the gap, which is really the normal process of the regular DNA replication. So then a good example of this process failing is melanoma. It's a type of skin cancer. So that occurs when this process of repair from UV light damage fails. The second type of repair mechanism, is base excision. And that is basically certain chemicals like nitrates. They cause deamination of DNA. In other words, it removes an amino group from the DNA. So it produces an enzyme called glycosylases, and that recognizes and removes the damaged bases. Damaged base. And then again, just like in the first one, it uses the DNA polymerase kicks in and it fills in the space, and then the DNA ligase seals the gap. The third final mechanism is mismatch repair, and that one fixes errors that occur during DNA replication. So it's not necessarily damage that occurs, it's more errors that occur during the process of replication, and that's called mismatch repair. It ensures that the fixing is happening to the correct strand, this one as opposed to this one. It knows because it has certain molecules that recognize where the damage exactly occurs. So it knows that, hey, the damage is in the daughter strand. Let's fix the daughter strand versus the parent strand. So now let's talk about the second repair mechanism, and that's called double strand repair. And again, as the name implies, it fixes both strands. It occurs when there's exposure to ionizing radiation, and the best exposure and the best example of ionizing radiation, of course, is X-rays. There are also three types under the double strand repair. The first one is non-homologous end joining. The second one is microhomology mediated end joining, which are very similar in the way that they repair the strands. But the third one is a little bit different, and that is called homologous recombination. Homologous recombo. So in the homologous recombination, a presence of an identical sequence is required as a template for the repair of the breakage. So let's see here, I'll demonstrate. So you have this chromosome that has damage, let's say somewhere here. So what this homologous recombination requires is a sister chromosome, the presence of a sister chromosome or chromatid right here. Oops. So this process right here, then it uses this sister chromatid to fix the one that's damaged. So this process altogether is very similar to a chromosomal crossover in meiosis. The rate of the DNA repair in general, using any of these mechanisms, it really depends on a lot of factors. Some being the type of the cell that's damaged, the age of the cell, and also the environment that the cell is part of. For example, a cell that's too old or had too much damage accumulated to its strands, or that isn't able to repair itself using any of these mechanisms, it can enter into a state called senescence. And it's sort of like a hibernating mode. It enters it's sort of a pre-death state. There's not much that's happening there. Another state it can enter when the fixing or the repair process fails. Another state is apoptosis. And this one is a sure cell death. That's what they call it. And then the third and last, final state, I'll just put it right here, is unregulated division, and that really is, a root of cancer. It's when cells divide outside of the actual controlled process of DNA replication. So that really leads to tumor growths, and they eventually do become cancerous. So this is how the DNA repairs itself.
As a result, the new DNA is created with one new daughter strand and one old strand.
You wanna? I should let you stand this way.
Oh, yeah, great idea. Okay, hold on.
And then you can like make the letters
Bigger, yeah.
Oh, I'm sorry.
Great idea.